Class, this is Dr. Ogilvy, and I'm reviewing Part C of the midterm exam. All the questions related to virtual slides. First question was identify the peripheral blood cell encircled. And I'm waiting for it to come up. And of course, this is a peripheral blood smear. You can see the red blood cells. You can see platelets. The cell circled is 1, 2, maybe almost 3 times the width of the red blood cell, and the average width of red blood cells 7.5 microns. So that cell is at least 20 microns in diameter, and it is the largest cell of all of the white blood cells. I don't see any granules in it. I see a very large nucleus. It's indented, and you have vacuoles in the cytoplasm against a kind of a pale bluish gray cytoplasm. So this is a monocyte. The next question was, in a normal blood sample, what is the highest percentage that would be represented by this cell type in a white blood cell differential count? And so, again, notice this cell is barely 1.5 times the diameter of a red blood cell, and so that would classify it as a large lymphocyte. But compared to the previous cell, a monocyte is much smaller, and this is a lymphocyte. And you notice it has just a tad of cytoplasm around it, and the highest percentage that you were given would be about 33% of the cells counted in a differential count. So now we'll pause this and go on to question 3 while I get it set up. Okay, for question 3, you would have to click on this radio button over here, and it brings up a field. You can see where the field is. It's in this specimen here in the reference image, and you see where it's located. But most importantly, you look at the pattern, and these are longitudinal sections of cells. I see that because they're like strings or wide strings, and there are nuclei, and the nuclei are in the center of these stained strands. And I can see here there's communication between this strand and this strand, and then I see these interesting structures here that are denser. So these are intercalated discs, and that's a nucleus in the middle of a striated cardiac muscle cell, or you could also say cardiac myocyte. So this cardiac striated muscle tissue forms the myocardium of the heart. The 3 layers of the heart: epicardium, myocardium, and endocardium. So now we'll go on to the next question. So for question 4, you need to click on this button, which will bring you to the magnification we wanted to ask the question about. Identify the round, almost empty space that contains a yellowish triangle. Well, you can look around here, and if you're a little bit confused, well, first of all, you're given this Masson's trichrome stain, and so you know trichrome, the collagen is either blue or green. In this case, it's blue, and the reddish area would be more calcified areas. So this is bone, and if you're a little bit confused, you could decrease the magnification and look around, and you see these patterns with an empty area, and you see multiple ones. So these are really Haversian systems, and the area here is called the Haversian canal, and it contains a blood vessel that runs through the bone, in this case compact bone. Okay, so we're going to go on to the next question now. So this question is asking, in what epiphyseal plate zone is the letter B located? So you're given the epiphyseal plate, and if you've got, in your memory, what the epiphyseal plate is composed of, that is a resting zone of cartilage, proliferating zone of cartilage, a zone where the cartilage cells are hypertrophying, getting larger, the zone where the cartilage is being calcified, and then the zone where a bone is being deposited on the calcified cartilage tent plates. So You might have needed to enlarge this. It's always a good idea to take a little closer look. And you see that these nuclei that are in lacunae in the cartilage are stacked in order on top of each other. And their mitosis is happening in one direction. These cells are dividing in this direction, and that's what's providing the increase in length of the bone. So this is the zone of proliferation. Now we'll go to the next question. So question 6, as we click on this button, the precursor of this collection of cells was a single cell. What is the proper name for this collection of cells encircled? Now the question is not asking for a specific cell type. It's asking for the name of this collection of cells. So you need to recognize the tissue. You're not told what the tissue is. But if you look here at this matrix, and these are the cells and the nuclei, the matrix is rather smooth and somewhat eosinophilic, but right around the cells, it's a little more basophilic. And so this should immediately remind you of cartilage. And if you decrease the magnification a little bit, you can see actually over here where the cartilage is forming a template, partially calcified, and there is where you're having bone deposited. So this must, and if you move down here, you'll see the cartilage is dividing in this direction. So anyway, this is a isogenous group, and all of these cells in here, all these chondrocytes came from one chondrocyte that was dividing. The next question is question 7. Okay, question 7 is identify the predominant tissue within the area outlined by the dotted line. And so here it is. Now, with my experience looking at this, I know this is immediately smooth muscle. But if I had any doubt, I would increase the magnification. And maybe by this step, I see now a pattern. I see little round nuclei here. And the nuclei are surrounded immediately by eosinophilic substance, which reminds me of smooth muscle, because in the smooth muscle cells, as you see in the longitudinal section, you have elongated nuclei, but the nucleus does not go the whole length of the cell. So you're going to have some cross-sectional profiles of smooth muscle cytoplasm with no nuclei. But when you catch it in the middle of the cell, you will see the nucleus. So this is the pattern of smooth muscle tissue. That's what you should have typed in. Now we accepted all kinds of little permutations of that as long as they're correct. At the end of the video, I'll just go through quickly some of the alternate choices we have made for a number of questions. Okay, so let's go on to question 8. Okay, question 8 asks about 2 cells are indicated by arrows. And you see, we've taken you to a high magnification within this whole structure here. And you look at these cells, the cells are round. That's one thing you can observe. The cells have a very large amount of cytoplasm, and it has a nucleus, and in the nucleus is a very prominent nucleolus. Now, I don't think you can magnify this anymore, but maybe I won't get in trouble. I'll just demonstrate something. I think you can, I was going to try to enlarge it. Actually, you can zoom in by doing this. But in this case, the way I have it and recorded, it's not enlarging. But anyway, this is enough to see that this is a large cell with a prominent nucleus, and the nucleus does not have hardly any heterochromatin. It's all very distended chromatin. So this cell is very active in producing something, which would have to be a protein. And in this case, it would be the protein, which is enzyme acetylcholinesterase. And this is a ganglion cell, and we would call it a sensory ganglion neuron. Now, I think we accepted neuron. We didn't actually require you to say sensory. But if you put ganglion, that is incorrect, because you have answered the question with a large structure and you have not identified the cell. So that's now here's a blood vessel with some red blood, and you can see the very thin, flat nuclei of the endothelium. And then here, this is these are nerve fibers that are running through this ganglion. Okay, so let's, and then around here, in fact, let's see, we're right here, these are satellite cells that surround each of these sensory ganglion neurons. Okay, this next question is asking us to identify in this specimen of the skin. You're told that this is skin. And immediately I can see that this is epidermis and this is dermis, but you may not be experienced enough to recognize that right away. But that's just what experience is. So in the specimen of skin, the blank is abnormally separated from the blank. Well, first you want to go over and enlarge this, and I want to find where there's a normal part of this specimen, and I'm spotting it right here because I can see here that here is the epidermis, and it's very typical of epidermis with the many layers of cells. And then this is an artifact where the stratum corneum is separated somewhat in this area. But as you increase the magnification, you can even see here the spaces around these cells, and I think that that's the highest magnification. So this is epidermis, and this is the first layer of the dermis, which is the papillary layer, and the collagen fibers are rather thin. It's rather loose connective tissue, and then here's the dense, irregular connective tissue of the reticular layer of the dermis. So if we move over here, we see the separation here is now occurring between the epidermis and the dermis. And you see the papillary layer of the dermis is right here, and there's the reticular layer down here. And actually, it's a pretty low magnification, but I can tell you that there are a lot of eosinophils here, and probably lymphocytes, and maybe even some neutrophils. So if we go back down to lower magnification, what this is a blister. And the water is accumulated in here and separated the epidermis from the dermis. So that should be your answers. So the next question is identify the structure represented by the thin basophilic line, and several examples were indicated by yellow arrows. And even below, and I'll click on that in a moment, you can see a cross-section of this. So I see a very thin basophilic line, and you know, in my experience, I just immediately recognize this as the axon in the middle of a nerve fiber, where surrounding it, the light area, is the myelin sheath. And now if I click on the cross-section, then that should be confirmed. And there it is. There's your axon, and then around it a light area, and that would be the myelin sheath, and out here a thin layer of the Schwann cell cytoplasm. And I'm not sure I will find it, but if you look around, maybe here is a Schwann cell nucleus. Maybe here is a Schwann cell nucleus, maybe there's a Schwann cell nucleus, and this is an axon here. So the correct answer to that, you would type in axon. If you typed in nerve or nerve fiber, you're absolutely correct. Nerve, you know, is a collection of nerve fibers, whereas a nerve fiber would include not only the axon, but the myelin sheath and the Schwann cell cytoplasm, the neurolema on the outside. So now the next question is question number 11. Okay, question number 11 asks you to identify the vessel encircled and use an internal structure or cell type to measure its diameter in the direction indicated by the arrows. Well, immediately down here I recognize hyaline cartilage, very dramatic example of the rather eosinophilic staining of the interterritorial matrix, and then the territorial matrix immediately around the cartilage lacuna, and occasionally you can see maybe a cartilage cell, but a lot of the cartilage cells have dropped out here. And then the chondrogenic perichondrium, where you see the chondroblast forming here, and then the fibrous perichondrium. But while looking at this vessel, we see red blood cells. And then what are we going to use for an internal measurement? Well, you could use the red blood cells, or you could use, say, a lymphocyte here, and it's about the diameter of my little indicator here. So I would just go 1, 2, 3, 4, 5, 6, 7. 7 times 5 is 35. Well, that's Very thin wall, no smooth muscle around it, not a capillary, certainly not a vein, not an arteriole, not any larger artery, and it is a venule, and if you put either venule or post-capillary venule, then it's correct. And this is the vessel that's just after the capillaries, and it collects multiple capillaries and then carries the circulation on. So let's go to the next slide, which would be next question, which would be question number 12. Okay, I've already clicked on the little button here to take you to exactly the area we're asking about. Identify the dominant specific tissue type within the box. And so you look here, and if you're at least a bit confused, you just need to look around here. You see these white spaces. You see a lot of mixture of eosinophilia, basophilia in this area. See a lot of eosinophilic staining here, and you see these holes. Of course, I immediately, it reminds me of compact bone, but that may have not been quite as obvious for you. So I'll look up here and we'll increase the magnification a bit. And when we do, we see that there are structures. In fact, I even think I see a blood vessel here and another blood vessel here in this structure. If you look on the outside, this is the edge, and at the edge I can even see here some osteoid and kind of a cement line where there's more bone forming here with osteoblasts along the surface. And so these are Haversian canals. This is compact bone. So that would be the correct answer. Not just bone, because there's cancellous bone and there's compact bone. And compact bone has Haversian systems, or the other term for the Haversian systems are osteomes. So this is a Haversian canal with a blood vessel in it. So let's go on to question 13. So question 13 now asks, what is the predominant cell type within the structure encircled? Well, you've got to recognize what these structures are, and so in your studies, this should remind you of nodules. And the nodules we've learned about really are lymphatic nodules. And then you have to think, well, you need to identify the organ just to know that this is a lymphatic nodule. Could be in the spleen, right? Could be in the tonsil, and it could be in a lymph node. This, in fact, is in the lymph node. And how do I know that? Well, I look down. Well, the medulla is not that obvious. And so I have to look up here and magnify this. And then I should see a capsule here. And then underneath that, I see lots of lymphocytes. And those are in the subcapsular sinus of a lymph node. And this is the nodule. And there you have a lot of dense basophilic staining and very lighter staining and more eosinophilic here. So this is a lymphatic nodule. And it's a secondary lymphatic nodule, and it has a germinal center. And in the germinal center, within here, the dominant cell type of the whole thing would be B lymphocytes, or you could put lymphoblasts or B cells, but not T cells. Because remember, T cells, they hang out deeper in the lymph node, in the deep cortex, or it's sometimes called the inner cortex. So B lymphocyte, B cell, B lymphoblast would be correct answers. Okay, now question 14 says, based on the histology of the cells in the field of this specimen defined by the dotted line, this specimen was taken from what organ? So now, your first impression is very eosinophilic with some blue spots, some basophilic spots, and they remind you of maybe nuclei. You increase the magnification, and maybe 2 times to the top magnification, and take a look. And you see these narrow strands. You see a nucleus in the center. It's certainly not a elongated nucleus like smooth muscle. Certainly, fibroblasts are not within collagen fibers, so this is not collagen. And then when we look around, we see this structure here, maybe another one here, intercalated discs, not too many. But you all see connections between the strands or between the cardiac myocytes, or this is cardiac striated muscle, and so it was taken from the heart. So heart is the correct answer. Question 15 asks, classify the tissue, the specific tissue type between the dotted red lines. Okay, now, the first thing I notice is that the nuclei are ovoid. And I also notice it's bordering a free space, and I see loose connective tissue underneath. So this has got to be some kind of epithelium. So I have to decide what of the 8 different types it is. Next, I look and see if the nuclei are all in pretty much one layer, and they are. If you look around, of course, you have occasional a variant here where maybe it looks like a nucleus. This could be a lymphocyte. This could be a lymphocyte here. But the dominant pattern is what you need to go by, is the nuclei are all in one layer. And so the cells are taller than they are wide. So it's very straightforward, I think. Simple columnar epithelium, not pseudostratified. Do not get confused by a minor representation of this whole tissue. Always consider the entire thing. In fact, we just outlined between these 2 dotted lines, so we were trying to steer you away from over here. Okay, so the next question is question 16. So question 16 is asking, identify the specific cell type indicated by the blue arrow in this cell, and ones like it make up a layer, part of which is defined by the dotted lines. So now some of you may recognize right away that this is the osteogenic layer of the periosteum. And right on outside, the more eosinophilic layer is a layer of fibroblasts and collagen, and that is the fibrous portion of the periosteum. But if you're not sure, then always look around. Well, over here, this is a skeletal muscle cell. You see the nuclei on the outside? Move around here. There's a slight longitudinal section of striated muscle, which is actually attaching here to the fibrous periosteum. That's really the way muscle cells attach. But if you move over here, you see now these nuclei in little spaces, and you see occasional little holes here that have a blood vessel in them. So this should remind you of bone. And the bone is compact bone. If you move over here, you move over here, you come to bone marrow. There's a lot of adipose tissue. This would be pretty much yellow bone marrow with a little bit of bone-forming cells. In fact, there is a megakaryocyte right there. So moving back across here to where we were asked the question and clicking on this is the osteogenic layer of the periosteum, and those cells are osteoblasts. Okay, so let's go on to the next question, question 17. So question 17 asks us to identify a cell type. 2 examples of the cell type are indicated by the black arrows. What does this differentiated cell type produce? So you first of all have to know what cell it is. And so with the adipocytes here, this large round spaces, and the multiple stages of cells being formed here, and then the very large and eosinophilic cell with a single but extremely large nucleus should immediately remind you of a megakaryocyte, and a megakaryocyte produces platelets. I think we accepted platelets singular and platelets plural. So the next question, when you click on the little button, takes you to this, and you're given that there's the lumen. Identify this organ. This specimen is a part of a cross-section of the organ. Carefully observe the tissue composition of the wall of this hollow organ. Okay, so we have to magnify this in order to identify it. So we're going up a couple steps in magnification, and we're going to look to see, but that's a lumen. That's a big hint that this is a hollow organ, and it's only a part of the wall, this hollow organ. But since we know this is a lumen, we're going to look for them inside and outside. We can see a difference in the tissue composition here you see a difference here, and then out here another difference. So I'm just going to look at, say, this region right here, and we're going to magnify this to the next level. Now I see very flat cells here, and those remind me of nuclei of endothelial cells. And then I see what looks like spaces between the nuclei and then some fine fibers, and this looks like loose connective tissue. Then I see these light pink strands here, and they're kind of wavy, and I can see multiple layers. I see one here, I see one here, and here, and here, and here, and here, and if I move along here, I can see multiple layers of those. And then out here, I see what looks like collagen, and I guess if I go at one more magnification, then those little thin wavy lines are very clear. And these are elastic membranes, not fibers, but membranes. And this is collagen out here. So this is the adventitia with some vasa vasorum, that is the vessels within adventitia. And then there's multiple layers of elastic membranes with some smooth muscle and fibroblasts between. And then here is the innermost elastic membrane. But it doesn't have just one elastic membrane, it's multiple ones. So that should remind you of the aorta, and this would be the intima, and of course the endothelium. So the next question is question 19. Okay, you're asked to identify the organ represented by this virtual slide. Even before I click, I'm not sure that goes, well, it goes to lower magnification. And if I increase the magnification, I see that there's actually two regions of this organ. There's an outer portion and an inner portion. So it reminds me of cortex and medulla, and this must be a solid organ, one with cortex and medulla. So what are the organs, excuse me, that are solid organs that have a cortex and medulla? Well, there's the lymph node and the thymus. Those are two that I can think of right away. And of course, you haven't studied, or you are studying right now, I think, kidney. It has cortex and medulla. But of the organs you studied before, you know, the heart doesn't have a cortex and medulla. Anyway, this is predominantly blue staining, and you see these rounded structures here that are on the outer layer. So this is the cortex of a lymph node and the medulla of the lymph node, where you see the secondary nodules with a germinal center. And then in the center here, you have the medullary cords and medullary sinuses. And the medullary sinuses here in this lymph node are just loaded with lymphocytes and probably some plasma cells that are going to enter the circulation by going out the efferent lymphatics in this structure. So again, if you move to the outside and move through the deep cortex, where you'll find most of the T lymphocytes, and remember those special vessels. Let's see, I don't know whether I can find one or not. I'm going to pause this and see if I can find a high endothelial vessel. Just a moment. So this is an example of a high endothelial venule. That is, if you look here, these are the endothelial cell nuclei, and they're not thin and flat. They're rounded. And if you move around, you can pretty much make out the endothelial lining. And there are lymphocytes, and there's probably a plasma cell with an eccentric nucleus, and some other lymphocytes here in the lumen. And you know, this is the place where the T lymphocytes that have been circulating in the body come out here and go into this deep cortex. So now we're going to go to the next question, which is question 20. Okay, question 20 says there are two vessels are indicated by the letters A and B. The upper specimen is stained with H and E, and the lower specimen is stained with elastic tissue stain. So we're just going to focus on the specimen labeled A. And so if we magnify this, and it's just part of a vessel, and it reminds you of a hollow organ. So you're thinking of heart, or you're thinking of blood vessels in this case, because that's all you had is the cardiovascular system that was hollow. And so if we move up in higher magnification and take a look at this, And we see some, even this magnification may be a one layer here, another one here, another one here. You look here, you have a very prominent positive staining for something here. And since it's stained with elastin, then it must be elastic membrane. So let's take a look, a little higher magnification at the inner portion at stained with H&E, which here you see this rather eosinophilic structure, and that's the internal elastic membrane. And if we magnify it one more step, and we can see that structure, and then throughout here you have smooth muscle cells scattered all in here. And then if we go down here to the elastic stain, then in the elastic stain you see you have a very prominent elastic membrane, and the rest of these are really fibers. You see they're not thick like this, so this is elastic fibers within the media of a muscular artery or a distributing artery, or you could say a medium-sized artery. So we accepted a variety of answers. As long as you didn't say aorta or didn't say venu or vein, you should get the answer correct. So we're going next to the next question, 20. So question 20, you're presented with this sample, which is very nice looking, I think. Very pretty colors of blue and dark blue and reddish, and different shades of red. And we ask you to identify the organ. So, you know, immediately when I look at this with my experience, I immediately know it's a spleen. I mean, I see, you know, cortex and medulla. I see a pattern of blue round structures, which reminds me of the lymphatic nodules in the spleen, which, by the way, are called splenic nodules. But we'll need to magnify this to be sure. So we just increase maybe 2 magnifications, and you can go up much higher than that. And you see these blue areas here. What you have to look for is, are they just simply, well, you know it's not the lymph node because you don't see a cortex and medulla. So now you look for one of the unique structures in the spleen, which is the central artery. The artery that runs along, and right there is a central artery. Here's one here and here. Here is one that's kind of longitudinal section. Here's another one here. And so when we look at this, and you see, look at it and see a central artery that has really no nodule associated with it, like maybe not this one. Here is a central artery with a nodule that's very dormant, non-active. It would be a primary nodule with no germinal center. But here is a central artery. It's kind of going on the longitudinal pathway. And around that, remember, that's the periarterial lymphatic sheath. And that's where the T lymphocytes are located in the lymph node. And in the nodules are where the B lymphocytes are located. Like here's a central artery, and here all these would be B lymphocytes. But when you're moving through the longitudinal direction of the central artery, where a lot of these are cross-section, but like in here, this would be T lymphocytes. Okay, so this is spleen. Let's go on to the next question, which is question 22. Question 22 asks you to identify the layer of this organ located between the 2 dotted lines. So again, you have to, I mean, if you look here at this reference image, you know, this is definitely a hollowed organ, and it's fairly thin wall, but you have to look. Here is an internal elastic membrane. There's the endothelium, and this is a rather eosinophilic cytoplasm with elongated nuclei, reminds me of smooth muscle cells. And then here you have the connective tissue on the outside. So this is the adventitia of a medium-sized or muscular or distributing artery. So the next question is question 23. So I already clicked on the radial button. It says identify the organ represented by this virtual slide specimen. So again, we have a predominant basophilic structure, but I can see dark areas and lighter areas. So with the basophilia and what I've studied or what you've studied in the midterm modules, you should be thinking of lymphoid organs, and you should be thinking of one with a cortex and a medulla. So how do you know for sure that which organ this is in? Well, what could it be? If the lymphatic organs, it has to be lymph nodes, spleen, thymus, or tonsil, right? And so you look here in the dark area, which should be the cortex, and you ask yourself, do I see any nodules? Well, I don't see any. I don't see any well-defined circular structures at all. So which of the lymph nodes does not have nodules? Well, there's only one, if I recall, and that is the thymus. So how do we know for sure this is thymus? Well, we just simply increase the magnification, and we're going to look for this unique structure, the Hassel's corpuscle. And so what are we looking for? We're looking something that's eosinophilic. Well, is it this? Nope, that is not it. That is a rather large venule that I'm looking at here. Right there. Red blood cells, maybe a lymphocyte or so within the cytoplasm. And we look around, well, this is beginning to remind me of a Hassel's corpuscle, but I'm going to have to go back down to lower magnification and maybe go to another area. And yeah, here, I'm sure I could look around and find better examples, but right there is one. And when you see it, I think you'll agree that this is a Hassel's corpuscle, and you see the whorls of the dying reticular epithelial cells that are forming this. Remember that the thymus has a reticulum, but it's made up of cells that are connected by desmosomes, and you can see these large nuclei here and there in the cytoplasm here of the reticular cells, like in here and here, and then these eventually, I guess, get worn out, and they just simply form this special structure. And remember, the reticular epithelial cells in the thymus produce thymosin. And the thymuses are not produced by thymocytes and not produced by the lymphocytes. It's a hormone that facilitates the transformation of naive cells from the bone marrow into T cells. So let's go now to question 24. Okay, question 24 asks you to identify the tissue or classify the tissue. When we say classify, there's epithelium, connective tissue, muscle, and nerve. Those are the four basic tissues. And then, of course, you have special tissues like bone and cartilage. You have subtypes of those. But if you're asked to classify a tissue, it's really the same as identifying, because we want you to go all the way. And we want, if it's epithelium, we want you to say if it was stratified squamous, non-keratinized, we want a complete answer. Here, in this case, you see all the eosinophilic staining should remind you of some kind of connective tissue, and very intensely stained with eosin. And if we look at this, we see a lot of eosinophilic structures that have no blue dots in them. The blue dots remind me of nuclei. And then when I increase the magnification and look at, yes, those are nuclei. I'll go one more step. And looking at this, I see what reminds me of collagen fibers. And everything is kind of organized in different planes. So this has to be dense, irregular connective tissue. Now, if we go back in lower in magnification, we see that this tissue is scattered throughout. And what this is mammary gland. And this is a mammary gland that is in a resting phase. It has no secretory units. All of these bluish areas are where the ducts are of the breast or mammary gland. And you're going to learn about that in Module 5, and these are all the epithelial cells of the mammary gland. And these form the ducts. And between those, you'll also have connective tissue that's a little bit more loose here with dominance of cell types. So this would be loose connective tissue here. And back out here where we were asked, you see this is loose connective tissue here between the epithelium, and this is dense irregular. Okay, so we're going to the last question now. Okay, I've already clicked on the button and it brought me to this where it says identify the organ between the two dotted lines. Okay, so now we have to decide what this is. Well, it certainly has different layers. There's a very dense and eosinophilic layer with some basophilic capping of that. And then you have these empty spaces surrounded by, it looks like, basophilic staining. And then you have very, very loose connective tissue down in here. Well, you know what this is really. This is the epidermis and dermis, and this is the hypodermis here, and down in here is really the fascia that covers the muscle, the interface between, if this is covering muscle. So you can't call this an integument because we're just restricting here between here, epidermis and dermis, and that forms the skin. So the best answer, and really the only correct answer, is skin. And if you want to examine this more carefully, you see this is the papillary layer of the dermis, is the reticular layer, and then you've got maybe a couple hair follicles here if we magnify one more step. And you could go on up to 20x and look at this very carefully. Here we have the very dense, irregular connective tissue of the reticular layer of the dermis, and you have the papillary layer, and then you've got your epidermis. We're going to go one more step of magnification, and you can see blood vessels now. You can see tiny capillaries here in the papillary layer. You see the stratum spinosum here and the stratum corneum. If we examine it higher, you could see the other layers. And then if you move down here, then we begin to see some of the glands, and these would be sweat glands down here. And actually, these are the sweat glands with the large lumen. These are like the apocrine glands that are found in the axillary region of our hypodermis, of the integument. But again, the correct answer to this is skin. All right, that concludes, and I'm going to now just go through a couple alternate answers we accepted just to give you an idea and give you good trust in us. Thank you. Okay, this question number 1, it doesn't show it here, but this was before I regraded, and we accepted momocyte, one letter off spelling. We accepted monocyte. And here we did accept 33%, and but if you gave anything other than that, I think, you know, we also, you didn't choose it, we accepted maybe 33, 34, 35. We're just trying to be a little flexible there. And then here, really the only, we accepted cardiac striatum muscle and cardiac muscle for this. And nobody, I don't think, put cardiac myocyte. For this one, we accepted Haversian canal, but then later on we went back and regraded for Haversian canal and Haversian canal and Haversian's canal. So we accepted all those. And then here we accepted proliferation, zone of cell proliferation, zone of proliferation, and zone of proliferation. We accepted isogenous for this, an isogenous group. We accepted smooth muscle and smooth muscle tissue. I don't think we accepted anything else for that. And we accepted neuron for this, sensory ganglion neuron, ganglion cell, and neurons, but we did not accept sensory ganglia. And if you put just ganglion, that's incorrect because that's a structure, and ganglia also. We accepted here either way, epidermis of dermis or dermis and epidermis. You could put either sequence, we accepted it. Here, the only correct answer here was the axon. And we did get a multiple attempts by you to get a correct answer, but all of those were incorrect. And I think if you pay attention to what I reviewed, and of course here's the postcapillary venial. We accepted with or without a hyphen, and you can see the exceptions there. And venial we accepted. Here we accepted osseous tissue, compact bone tissue, and lamellar bone. But if you put cancellous, which nobody did, So we were pretty lenient with this. We should have just perhaps stuck to compact bone, but we gave you the benefit of the doubt. And here we accepted multiple answers: B lymphocyte, B cells, B lymphocytes, B with a dash, etc. You can see the variety of answers we accepted. And then on the heart, there's only one answer for that one, and that was heart. And here we accepted simple columnar epithelium, various permutations of that, but not pseudostratified columnar epithelium. If you just put epithelium, that's wrong. You've got to be complete. And then the osteoblast or osteoblast, singular or plural, but nothing else for that one. And then, of course, these cells, megakaryocytes, produce platelet, or you could have said platelets. And question 18, a large elastic artery, aorta, accepted that, large artery, elastic artery, and aorta. And that's all we accepted on that one. And of course, this was very simple. Now, somebody put lymph node with no space. We accepted that. And then for the medium-sized artery, distributed artery, and see, this is misspelled by one letter. We accepted that. Medium muscular artery, medium muscular artery. And then here we accepted a misspelling, one layer of the spleen. And here, different layer. We got tunica adventitia, sometimes misspelled by one letter. So there are a variety of answers we accepted there. And then the thymus, only one answer for that. Question 24. Dense irregular, we accepted some misspellings for that. And then here we have skin. And we did accept thin skin, but nothing else. Not integument. That is not the integument. We ask for the skin. Okay, so that includes this. I hope that was helpful.